In this section, we're going to look at the components that are suspended within blood. So let's go down here first to this scene. So here we have a phlebotomy taking place where we're going to do a blood biopsy or simply take blood out of a vein and then put it in a centrifuge for about 15 minutes. And then once the centrifuge is done, this is what the blood is going to look like. It's going to be broken down into plasma, the buffy coat, and erythrocytes. Okay, plasma is all the yellow stuff that you're seeing there, and then you have the buffy coat, which is filled with leukocytes, which leuko means white, cyte means cell, so these are your white blood cells, and we're going to look at 5 different ones here in a little bit. And then your platelets are also found in the buffy coat. All right, going back up here, let's look at the white blood cells. So we'll start with that. Okay, a white blood cell or a leukocyte are going to be the, in order, neutrophils, lymphocytes, monocytes, eosinophils, and basophils. Okay, now this is in order from most abundant to least abundant. Okay, there's a mnemonic that goes with this that would help, and it would be never let monkeys eat bananas. Okay, now if you can remember that, never let monkeys eat bananas, what you're after is the first letter of each white blood cell. So the NLM, E, and B. So never let monkeys eat bananas. And that's how you remember them from most abundant to least abundant. Okay, the other way to remember them is granulated versus agranulated. Okay, when you add the prefix a to a word, it means without. So the agranulated would be without granules, and the granules are the freckles that you see on these white blood cells, and there's 3 of them. So the granulated one are what I call the bens. So nothing but the Benjamins or the bens. And that would be basophils, eosinophils, and neutrophils. All right, so a basophil, you can see the granules, and we'll talk about what the granules do here in a little bit. And there's the eosinophils and their granules, and then you have the neutrophils and its granules. Okay, I prefer you look at the mature neutrophil because of the different lobes that it has. We'll talk about those lobes in a minute. Okay, so to recap, the white blood cells, you can list them in most abundant to least abundant. You can also list them as granulated versus agranulated. Okay, the agranulated ones are the remaining 2 of the 5. Okay, so we've got 3 granulated and then 2 agranulated. So that leaves you with the lymphocyte and the monocyte. And if you take a look at the monocyte, you'll notice there's no granules. And if you take a look at the lymphocyte, you'll notice in the cytoplasm there are no granules. So therefore they are agranulated. Okay, the other cells that you see in here are going to be the red blood cells, also known as erythrocytes. Erythro means red, and cyte means cell. And then you have these tiny little dots. Those are the platelets. And the platelets' job, or one of their functions, is coagulation or healthy forms of clotting blood. And another function are growth factors. So there's over 35 different growth factors found in platelets. And this is where we actually get the technique of PRP injections, so platelet-rich plasma. And what you do is you concentrate down the plasma, because what you're after are the platelets that are suspended within the plasma, and then you inject it into the site of interest. So, for example, the tendinitis in the elbow isn't going away, or there's a disc injury, a vertebral disc injury. Then you inject the PRP into that injured site, and those growth factors they kind of behave like stem cells in the sense that they will do some growth and repair. Really good technique, the PRP. Let's look at what the functions of the leukocytes are. Okay, so we'll start with the neutrophils, let's do the granulated. We'll do the granulated ones first. Okay, keep in mind, granulated are the bins. So basophils, eosinophils, and neutrophils. Okay, the neutrophils, they are 3 to 5 lobes. That's one way to identify them under a microscope. Okay, so they have 3 to 5 lobes, and they are also the first responders, and they have granules. Okay, so looking at the neutrophils, they are also known as phagocytes. Okay, so the word phagocyte means that it has the ability to destroy a pathogen. So I like to use the Pac-Man analogy. So Pac-Man goes around and eats things. So the neutrophil goes around and eats things. So that makes it a phagocyte. The actual process of destroying the pathogen is phagocytosis. Okay, an example would be bacteria. Okay, the granules that are inside of the neutrophil are going to release things like enzymes. Okay, an enzyme in this case is going to help break down a variety of proteins on the pathogen. It also helps with inflammation. So the inflammatory process actually brings in other white blood cells to help. Okay, then it also helps prepare the area for tissue healing. Okay, the eosinophil, which is granulated. Okay, so we'll look at the eosinophil over here. It is bilobed. Okay, so you'll notice that there are two lobes. And the way we get these lobes, or the reason why the lobes look purple is because of a dye, or you could say a stain. So they stain them, the cells, so that they're easier to see under a microscope. Okay, an eosinophil, which is bilobed, it's going to help us with an allergy response. Okay, now we're talking about a normal allergic response, and you're probably like, well, what the heck's a normal allergic response? Well, when people say allergies, normally what they mean is hypersensitivity, hyperallergies. It's too much. So they take Claritin or allergy shots. But under our normal defense system, if particles and things get inside of us, you actually want to snot and sneeze and all that allergy response to get it out of your system. Okay, but obviously people can cross the line where you get into hypersensitivity and hyperallergy stuff. Okay, the other thing that eosinophils do is they help kill parasites. So on a blood lab, or a CBC, which stands for complete blood count, if you see elevated eosinophils, then one thing that you can be thinking clinically is this person either has an allergy response going on or they're fighting parasites. Okay, the next one under granulated would be the basophil. Okay, would be the basophil. And that one that's the least abundant, but it also has a lot of granules in it. All right, the basophil granules release histamines. Okay, they release histamines, which is a vasodilator. And if this spins out of control, people can take an antihistamine, which essentially will decrease the diameter of the blood vessel. So vaso is blood vessel, dilator is opening up, and basophils also release heparin. Okay, heparin is an anticoagulant. And these two things, the release of histamines and heparin, are taking place during an inflammatory response and or during an allergic reaction. Okay, now we're left with the A granulocytes. And we'll start with the monocyte. Okay, and here's what it looks like. So there's your monocyte. They are big. They're actually known as the big eaters, and it has a kidney-shaped nucleus. Okay, when a monocyte so there's a monocyte that is inside of a blood vessel. When it leaves, it turns into a macrophage. Okay, so the reason why it would leave is because the site of interest, okay, so we'll call that X, means injury. So there's bacteria and all this other stuff. And a macrophage knows to go to this area because of a chemical signal called chemotaxis. Okay, chemo means chemical, and taxis means movement, like a taxi cab or taxi. So chemotaxis is chemical movement. So the bacteria or pathogen or whatever releases a signal. It does have its own chemical signal, and the white blood cells are attracted to that. So the macrophage knows to then go to the site of interest. So the actual leaving of the monocyte from the blood vessel is called diapedesis. Okay, there are little holes, little pores, okay, little holes inside of our the blood vessels so that things can actually leak out. So diapedesis is the process of a white blood cell leaving the blood vessel, going to the site of interest. A monocyte, when it leaves, so diapedesis, turns into a macrophage, goes to the site of interest, and does its engulfing. Okay, so it's a big eater. It is a phagocyte, and it also cleans up dead cells and debris, and it's also responsible for a major histocompatibility complex, which is part of our immune system, which that's a discussion for another time. Okay, then the last one is a lymphocyte. Okay, so it is agranulated. So you can see the cytoplasm does not have granules, and you see a nice big nucleus. It's also the second most abundant white blood cell. If you go back to the never let monkeys eat bananas, so the N and the L, so the L would be lymphocyte, so the second most abundant. Okay, so a lymphocyte is going to produce antibodies. Okay, those are Y-shaped proteins, and that is a very powerful part of our immune system, part of antibody-mediated immunity.